I didn't do that.
Oh, thanks so much, Emily. I appreciate that.
Yeah. No, we left. We left because we had to come here. And Rasmus was still.
Rasmus was asking about 500 questions.
Went to my advisor meeting.
Yeah.
Sat down and she goes, okay, well, all you have is 3 classes left, so the rest one is elective, so you just pick whatever you want.
So now, nice and easy.
Yeah, but I'm gonna ask Martin, because I was talking, I was like, do you think like a BS is worth it? And she's like, for geology, because it's surveying, she's like, for geology, yeah, it makes a difference, but for bio, I think that, like, when they taught us, like, what they told us was like, one doesn't really matter. Orgo and genetics lab.
I thought I would be having a
So what I heard with environmental science and bio, the BS.
Yeah, that's what she told me.
Oh, is that what you said when you're?
Well, if I was going to high school, I would already be doing like shit, at working at that's I'm already behind all that stuff.
You're good. Yeah, we need to consider it pretty awkward. I'm not looking at the yacht, I'm already.
My brother did the TS and he's gonna be a professor now.
So when you're going into that school, are you just going to tech, or are you going to try and
I'm
get your
going
own?
to school that surgeon.
So you're going to try and get your own practice?
Big plus.
Yeah, someone like, and then like, it's all like,
Pretty stressful, yeah. Like Martin, he's my advisor. He said I should switch to
Just go.
I already took all that stuff though, like all the distribution stuff.
Yeah, I haven't done my general stuff. I've done all my final stuff. I'm done with all my final. I just haven't done like
Yeah.
And so he handed me the new cataloger. He had Rasmus sent to me and was like, okay, here, figure out what you need to take and kind of because he's getting confused. He doesn't know what he needs to.
It's just all GCOM stuff. I'm taking GCOM. Yeah, GCOM like that's for my religion, and that's the last one I need.
I have
Everything
GCOM
else.
120 now. The rest I don't.
All my stuff is, all the stuff is just like random stuff. I literally took from my art because I took Photoshop.
That's what my brother does.
I just don't know, like, I honestly have to find like.
I'm the one.
I was looking at environmental science lessons, and I don't have, like, something interesting. Only the bio, though, this is like conservation bio. And then they have like exploratory internships and special internships. So I don't know. I have like a big internship.
Was that my
Yeah.
He's in the golf program and like he's going on internships like all next semester.
Oh, yeah. I think you're telling me that you're not excited.
I just gotta be.
Yeah. Sure.
This is 2024, 2025, so.
Even my he's new kind of.
Where do you find that at?
We should schedule a meeting next week. We can talk about this.
No, I think you should bring down here, you go here.
We should schedule a meeting next week to talk about this.
I don't think that comes with my head.
The academic catalog?
It just looks.
That's not coming out like being bitchy, but if it does come out, then like, I know I didn't do that. Like, be coming home and I talked in the phone here, but you can like, you can do that. No, but I know you're here. Yeah.
This is all the left.
Even though I don't know. Talking about I don't know.
And the rest is just,
Everybody's from home. That's that's why I was like, what?
Because I thought if you both my senior researches, the same semester, second first semester.
Oh, yeah.
I'd rather.
Well, that's why I was my one friend. It's like, I don't know. You like call like once a month. And we're like, great. Like, okay.
Well, happy Friday, everyone.
Happy Friday. We made it. Another week.
Another week.
Yeah, close to the end.
So today we should be able to finish chapter number 14. So there's a few more things to discuss. And don't forget about that practice exam that's posted on Blackboard. And also don't forget about our exam that we have scheduled for Monday during class time. So also don't forget about uploading your notes. So upload your notes when you're writing. Yeah. Okay. I guess any questions about the exam coming on?
It's Monday, not Friday. I thought it was next Friday, it was Monday.
Well, I mean, I think I don't have my calendar with me, but we're coming up to Easter, aren't we? So,
I think next Friday.
Yeah,
Good Friday.
so there's no class Friday, right? On Good Friday? Okay, so.
It's Monday, yeah, I gather. Like you said something, I wouldn't have studied at all.
Okay. All right, so let's begin by taking a look at this overview to see where we are at. So last time we learned about interferon. So we're actually over here on the panel on the right here, looking at different what they call effectors. So things that could help to slow or eliminate the growth of potential pathogens. So today we're going to look at the complement activation. How do these complement proteins affect growth of microbes? We'll look at phagocytosis, inflammation, fever. So those are the 4 things left to learn from chapter number 14. All right, so let me just kind of skip forward a little bit here. So let's see here. Here we are. All right, so this is a really nice graphic that kind of shows the 3 different actions of these complement proteins. So these are there's a number of them that are labeled by the letter C and a number, like C3 and C5. And then when you see the letters A and B following those, it just refers to pieces of one of the complement proteins. So like C5A is part of C5. Okay, so let's kind of focus on these 3 graphics here. Next slide will actually give us a whole bunch of words that describe what we see on the slide here. So the one on my right here, or on your right, is called opsonization. I know it's you have to kind of squint, but we'll see it printed much larger on the next slide. Opsonization. So here we see that some of the complement proteins, like the C3b, for example, can bind to a microorganism like a bacterium, and that will actually flag that microbe as being a foreign object that needs to be destroyed by a phagocyte. So that's what we're looking at in purple here. So this is a cell, one of your immune cells that will engulf and destroy, internalize and destroy that microorganism. So these complement proteins, through this process of opsonization, bind to that protein, or bind to that microbe, and then basically, in effect, make it more easily internalized by that phagocytes. So you can actually see that the phagocyte actually has specific receptors that will bind to the complement protein in order to facilitate that phagocytosis process. Okay, so this is one possible outcome, one benefit of having these complement proteins, and that is the digestion of these microbes by your phagocytes. Okay, so we call that opsonization. Moving down below, we have lysis. Lysis of foreign cells, so that could be like a bacterium, by the formation of what they call a membrane attack complex. So that membrane attack complex is represented by these purple and yellow kind of oblong structures here, each of which is a complement protein. So this structure here, the membrane attack complex, is formed by when we have an aggregation of these complement proteins that will actually form a pore. So you see that this structure looks like a cylinder, and then because the pore is formed, that will allow the cytoplasm of that bacterium, so the bacterial cell would be down below, that would leak out through the membrane attack complex, and that would lead to the death of that particular bacterium. Okay, so this membrane attack complex puts a hole in the cell, and will lead to the eventual death or lysis of that cell. So that's a good thing for us too. And then over here on your left, we have inflammation. So some of these complement proteins are involved in enhancing or facilitating inflammation, which is a dilation of your blood vessels here that will allow increased blood flow. So that will bring more immune cells to the site of infection, and then dilation will also result in increased permeability, meaning that those immune cells can exit out of those vessels more easily if they're more permeable, and then they'll be able to access, basically attack those pathogens that may be present nearby. So again, those are the three. Next, we're going to see a bunch of slides with just words that parents describe what I just said. But I don't know. I like pictures. All right, let's see the words here that go along with the pictures here. So again, we're looking at three outcomes here, three possible actions of these complement proteins. First one was opsonization. So I know that one's something that you don't see every day, right? that word every day. So again, binding of the complement proteins to the bacterium will actually promote the destruction of those bacteria by phagocytes. So those phagocytes could be your macrophages, they could be your neutrophils, they could be your dendritic cells. So whatever cell you have around there, that's a target. That makes it more likely to be destroyed. Next on this list is inflammation, or the inflammatory response. So again, we have dilation of blood vessels, increasing blood flow, more immune cells, travel to the site of infection. Increased permeability means that those immune cells can exit out of those vessels and then reach the potential pathogens. So that's all facilitated by complement proteins as well. And then the last one on this slide are the membrane attack complexes. We saw where those yellow and purple oblong shaped complement proteins form a hollow cylinder that creates holes in the membranes of bacterial cells, causing their contents to leak out and eventually leading to their death. All right, let's look at some more. I like pictures. Let's take a look at this picture here. What are we looking at? Super hard to tell because these things are so tiny, but this is actually what these arrows here are actually pointing to membrane attack complexes. So here's our membrane, the plasma membrane here, and then we see these tiny little holes. Tiny little holes, right? Formed by those complement proteins that could cause cytoplasm to leak out. Okay. Wow. You need good eyes. Right? All right, any questions about this slide or the slide before? Three possible results of these actions of the complement proteins that are part of your innate immune system. All right, next we're going to learn about this process of engulfing and destroying potential pathogens we call phagocytosis. So we actually have a number of steps here that are kind of shown in these rectangles. So we'll start off with the first one here, referring to called chemotaxis. All right, we actually saw that word before. Who can remember what does chemotaxis mean? Movement.
Okay.
Directed by chemicals, that's right, yeah. So, in this case, often it's an attraction facilitated by chemicals. So we have binding of the complement protein C5a to the microorganism. So presumably the exterior of that microbe has chemicals that will attract the binding or facilitate the binding of these proteins to it. So that'll give us this kind of a picture down here. They switch colors on you, but this is what the microbe would look like after the complement proteins bind to it. Okay, so that's chemotaxis. Step number 2, recognition and attachment. So recognition by, can anyone figure out by what? What is recognizing this? Okay, here's the chemotaxis. And then recognition, attachment of this by what? Think of something else. Can you figure it out from the graphic? What's this purple thing? That's a phagocyte, that's right. So recognition and attachment by the phagocyte. So that's what this purple thing here is. So you see this binding reaction taking place here between the receptors of that phagocyte and this upsized complement attached covered bacterium here. So that's the recognition and attachment occurring here. Okay, so let's go on to number 3, engulfment. Okay, so that's when the membrane of that phagocyte covers that microorganism, and then ends up internalizing it. And then number 4, the phagosome maturation. So we have internalization of the bacterium within a vesicle. So that'd be like it's covered by the membrane that used to be on the outside of the phagocyte. Let's see, number 4, destruction and digestion. Okay, so we're looking at this picture here. What are these little circles here that have the little dots inside of them? Can you see what those are? Those are the lysosomes, and inside of which contain those lytic degradative enzymes. Okay, and then when the lysosomes fuse with that phagosome, they release their contents inside, and then we have breakdown of the bacterium into smaller parts. So that's the destruction. And then exocytosis. It's like the opposite of endocytosis. So we have fusion of the phagosome to the membrane, and then the contents of which are released outside. Okay. Now, some of those contents could actually end up in the membrane, so let's keep that in mind too for the future. Okay. Any questions about phagocytosis? So you have phagocytes that basically, this is kind of like an eating, if you think about an eating process of internalizing it, digesting it, excreting. If you think about it. Okay, and then, so we saw the graphic, then we're going to get a bunch of slides with words on them. This slide informs us that the macrophages are one type of phagocyte, and they do respond to cytokines. And there's different kinds of cytokines out there. Remember, that's how these cells communicate with one another, by releasing chemical substances from one cell that are received by another cell. So these cytokines can actually activate these phagocytes and possibly bring them closer to where the infection is occurring, where the microbes are, in order for them to function. Okay, so that's the idea here about the cytokines. One type of phagocyte is a neutrophil. Remember, neutrophils are granule-cytes. They have granules on the inside of them. The granules are those lysosomes. So the ones that have the lytic enzymes, the degradative enzymes. Okay, a little bit of information about the neutrophils. They are one of the first to arrive at a site of infection. So remember, you got a bunch of different kinds of cells, a bunch of white blood cells, but the neutrophils are the first to arrive. And they are phagocytes, so they will go and engulf and destroy. Okay. All right, next we're going to learn about the inflammatory response. We're looking at four characteristics of inflammation. We have redness, Why does a site of inflammation turn more red? More blood, right? More blood is moving into the area, right? Remember that we have blood dilate, the vessels are dilated here. Okay, and why would that cause swelling? More volume, right? Because the vessels are dilated, more volume moving of liquid and cells moving into that area, that causes swelling, heat, and also discomfort, pain. Okay, remember, is pain a sign or a symptom? Symptom, right? So, like it's difficult to tell how much discomfort or pain people are in, and then you can try and quantify it. Like tell me on a scale from 1 to 5, how much pain are you in? And you tell me 10. Okay. Inflammation is a good thing. Look at this here. The idea is to try and, you're bringing in your immune cells in order to eliminate the pathogens, keep the pathogen from spreading. So that's what they mean by localized versus, anyone know the term for when pathogens spread to other parts of the body? Starts with the letter S. Localized versus systemic. Very good. So you don't want spread. So systemic is when things spread. Okay, and then once the pathogen is removed, then you have restoration back to normal tissue function. Okay, so yeah, a lot of times when you see inflammation, people are thinking, oh, that's bad. Well, it's actually beneficial. Okay. Yeah, what promotes inflammation? Well, we have histamines. Anyone remember what granule site releases the histamine, or can release histamine? The basophils. That's right. That's their job, and so that's actually helpful. But then if you have too much histamine, then that's too much of anything is not good, right? Okay. And then here's a listing of different kind of phagocytes. So which includes the neutrophils. The term diapedesis refers to when those phagocytes squeeze out of the blood vessels, and then they're able to encounter and eventually destroy the pathogens. So that's the diapedesis. Remember that our cells don't have a cell wall, so they have that flexible membrane. So that means they can squeeze through pores if need be. The myelocytes are the precursors for macrophages and dendritic cells. And then in addition to these cells, you also need to, kind of heal if there's like a puncture or something, depending on what the wound is. So it's nice to have some clotting factors there as well. Okay. This makes more sense with the graphic that's actually coming up here. So let's take a look at that one next. Okay, so we're going to look at the steps of the inflammatory process beginning on the left here. So, oh my gosh, you get stabbed. I don't know, what is that, a knife? Or a needle? Something. A nail? And what's even worse is that it's covered with bacteria. So now your skin, remember that's a barrier to infection, has been breached. And now you're exposed to harmful bacteria. So that's our first panel. But then we have, the inflammation starting off here. So again, that's, the vessel will dilate. And here's a picture of the diapedesis here. See how the cell can squeeze through a tiny little space in order to exit out of that vessel and then be directed to where the microbes are. So, what kind of cells are we looking at here? Neutrophils. Neutrophils are the first to arrive. So that's a type of granule site. So here's an example here. And the granule is that lysosome, which has the lytic enzymes, the degradative ones. Neutrophils are phagocytes, so they will engulf bacteria. And then let's see here. There's always there's going to be cytokines released that will attract additional macrophages. So it's not just one or two neutrophils there. You'll have additional macrophages, and they will work together basically to eliminate the microorganisms there. And then this slide didn't mention those clotting factors because there's that puncture wound. It'd be nice to heal up the blood flow, prevent the blood loss. All right. Do you like that diapedesis? That's pretty cool. You wish you could get skinny like that sometimes? Squeeze through small openings? All right, we're actually getting near the end here. Any questions about this one? Okay. Now, ideally, our immune system is working very quickly to address and potential and prevent infections. So, ideally, the inflammatory response is short in duration. So, we call that acute. Acute meaning brief. But then, there's, sometimes things don't work as intended, and the inflammatory response continues for a longer period of time, and then we use the word chronic. So I think we've actually seen these terms before, when we were talking about different kind of diseases. So we're just applying them to inflammation. So again, acute refers to short duration, chronic, over a longer period of time. So we, worst case scenario, it could be years here, years, multiple years of inflammation. Well, why would that be? why would we have chronic inflammation? Our immune system is designed to identify foreign substances, that are not supposed to be, part of your body. So those could be potential pathogens. They could also be allergens. So they could be non-living entities like pollen, dust, whatever. But then sometimes our bodies have a hard time identifying, normal cell versus a foreign cell, and we'll make the mistake of attacking our own cells. So that is referred to as an autoimmune disease. So auto meaning self. So your immune system is recognizing your own cells and is triggering a defense response. So one example of that would be rheumatoid arthritis. So you may have maybe have some older relatives who are who have some joint pain. It's common in like hands, for example, and that may be due to an autoimmune response where their immune cells are actually attacking the cells and tissues at certain joints. And that could be over a long period of time, so hence the term chronic. Now, interestingly, scientists are interested in looking at using our immune system to treat cancers. Because those cancer cells, because they become mutated, they are different from your normal cells. So why not employ your immune system to try and eradicate, use those those phagocytes, for example, to engulf the cancer cells. But at the same time, those cells are still your cells, so it's really hard to tell what's foreign and what's not foreign there. So scientists are working on that, but it's not an easy one to resolve. All right, we're near the finish line here, so hang in there. All right, we have a couple more terms here, we've seen this word before as well, apoptosis or apoptosis. We saw that when we learned about the interferons, where the cells that are infected with the virus undergo apoptosis, then that will prevent that virus from replicating and will, as a form of self-sacrifice, eliminate the infected cells so that the uninfected cells can survive. So that, and if you have a whole bunch of cells dying, then we have a term called necrosis. Or like necrotic would be like the adjective if you have an area. Hopefully you don't have it, but there might be an area where you have a bunch of death, cell death, tissue death happening, and then you hear the term necrotic. Now, necrosis could be caused by apoptosis, but it could also be just caused by the pathogen itself causing cell death. Last slide. You did make it to the end. we talked about how inflammation, even though we think of it as a negative, it actually has some positive benefits. Having a fever is, we can think of it as a similar way that it's actually a normal part of your defense systems. There's actually benefits to having an elevated temperature. So, and that's related to the temperature preferences of bacteria here. Like we learned that bacteria have preferences for their physical environment in terms of things like oxygen requirement, nutrient requirement, carbon source, so on. Well, that also includes temperature. They have preferred temperatures to grow. So the idea with fever is that our bodies are elevating the temperatures above which the microbes prefer to grow. So that will slow them down. And at the same time, these elevated temperatures actually will help activate your immune response. So it helps with inflammation, bringing in those phagocytes. The phagocytes get even more hungry, more active, and will destroy and consume pathogens faster than before. Your lymphocytes start multiplying, dividing, get more immune cells, more granulocytes than before, and then we're going to learn in the next chapter more antibodies. Again, too much of anything is bad, right? So if you have elevated temperatures, if it's too high, or if you have fever for too long, that could have negative consequences as well. But a mild fever is a good thing. Okay, so, boy, I'm hot right now. Aren't you hot in this room? My immune system is happy. Your immune system is happy. All right, any questions? Okay, just a reminder, we do have our next exam scheduled for Monday. There's a practice test online on the Blackboard site. I'm recording everything, so if you want to review the recordings, they are there. PowerPoint slides are also online. And did I mention practice test? Practice test is online too. All right, so have a great day, everyone.